Hello and welcome. Today let's talk Chapter 4. Chapter 4 is going to take us into membrane structure and function. So today we'll focus more on the plasma membrane. We'll look at the structure of that plasma membrane, see how it's built, and then we'll look at the function of that membrane. We'll see all the components and basically what they do is what we're going to check out. So here when we start with 4.1, we talked previously about the plasma membrane, and we talked previously that the plasma membrane is going to be found separating the internal environment of the cell from the external environment. Now, along with this, it's going to regulate the entrance and the exit of molecules. It regulates the entrance and the exit of molecules from the cell. So to regulate the entrance and the exit of molecules from the cell. You'll see that this is going to help the cell and the organism to maintain a steady internal environment. Or we can say it helps the organism to maintain homeostasis. Maintain a steady, stable internal environment, or homeostasis. We said before that the plasma membrane is made primarily of phospholipids. We had a phospholipid bilayer. Cholesterol is also going to be found. We'll see it's a phospholipid bilayer, and it's going to be embedded with proteins. So there's going to be proteins embedded into it. And we're also going to have cholesterol found in relation to it. Cholesterol is going to be found within the membrane, and it serves to stiffen and strengthen the membrane. So proteins are also found in numerous quantities. Proteins are going to be found in numerous quantities, and they're going to be found scattered basically on the outside and the inside of the membrane. Proteins are also going to be found in numerous quantities, scattered just outside or within the membrane. And these proteins can be found either partially or wholly embedded within the membrane. Now these phospholipids, we looked at the structure and we saw they had a hydrophilic and a hydrophobic end. The hydrophilic end is going to be the water-loving end, and that's the polar heads. So the polar heads are hydrophilic, and they're going to face inside and outside of the cell where there's water, because again, they are hydrophilic. When we look at the tails are nonpolar. They're hydrophobic, water-fearing, nonpolar tails. And they're going to face each other, and that is away from the water. And then again, cholesterol there also. So let's talk more about the proteins. Now we talked membrane proteins, so they're found throughout the membrane, and they may be peripheral proteins, so this is one type. And these peripheral proteins are going to be associated with only one side of the membrane. They're associated with one side of the plasma membrane. We also have integral proteins. Integral proteins are going to span the membrane. They're going to span the membrane, and they can protrude from one or both sides. They can protrude from one or both sides. And they're embedded within the membrane, and they're able to move laterally. Now together, the combination of the proteins, the steroids, the phospholipids, this all gives this plasma membrane, this fluid mosaic model name because of the appearance. So we call this a fluid mosaic model. Beautiful structure. Here's all the phospholipids, and we said phospholipid bilayer. So that's one layer you see right out here on the top, and then the other layer right down here, it's a bilayer. Phospholipid bilayer, two layers of phospholipids. And here's the polar heads, because there's extracellular fluid on the outside, intracellular fluid on the inside. And then we can see here, there is the tails, the hydrophobic tails, nonpolar tails. You see all inside of here. They have a little kink in them. We talked about that, right? We saw the double bond created the kink. And we looked at the structure there as well. And here's the proteins. So here's some integral proteins. Okay. And then here you can see you've got some peripheral proteins there as well. So very nice structure. We'll look at this in greater detail. Now, when we talk about the function of the membrane proteins, okay, so here we went through a couple of those proteins. Now, I want you to go through, we'll talk about the function of these proteins, but there's phospholipids, and proteins can have attached carbohydrate chains to them as well. You can see that right inside of here. Here's an attached carbohydrate chain to this protein here, and then here you can see also to the phospholipids. So both phospholipids and proteins can have attached carbohydrate chains. And glycolipids are lipids attached to carbohydrates, and glycoproteins, proteins attached to carbohydrates. So glycolipids, we see some in here, okay? And then glycoproteins, we see there. And then here, we can see good old cholesterol in purple. Cholesterol right in here as well, and here too. So let's talk function of these membrane proteins. Now, we talk about the function of membrane proteins. The peripheral proteins have a structural role, and they help stabilize and shape the plasma membrane. The peripheral proteins have a structural role, and they help stabilize and shape the membrane, the plasma membrane. And they may also function in signaling pathways, and they may also function in signaling pathways. The integral proteins can be of the following types. We can see here we have first channel proteins. When we talk channel proteins are involved in the passage of molecules. They're involved in the passage of molecules through the membrane. They have a channel. They have a channel that allows a substance to simply move across the membrane. Channel proteins may even contain a gate that must be opened then, and that will open by the binding of a specific molecule to the channel. So here we can look at a nice picture of a channel protein. So these channel proteins, we say, are involved in the passage of molecules through the membrane. So these are molecules that can't just pass right through the phospholipid bilayer of the membrane. So since they can't pass through the phospholipid bilayer of the membrane, we have special channels that are going to allow them to pass, and these channels are proteins. So proteins, you can see, aren't just for muscles and these few things there. We're going to talk, when we talk more about proteins, about their function, and we'll check that out as well. So here next, then, when we go through and we talk channel proteins, so they're going to allow, we said, passage of molecules through the membrane. They have a channel that allows a substance to simply move across the membrane. These channel proteins, I told you, may also contain a gate. They can also contain a gate. And that gate then has to be opened, and that's going to be opened by the binding of specific chemicals to the channel. Next, we have carrier proteins. Here we can appreciate a nice carrier protein. Okay, so you can come back here and you can kind of read the text from there as well. So when we talk carrier proteins, they are also involved in the passage of molecules through the membrane. They're also involved in the passage of molecules through the membrane. However, now these proteins are going to actually combine with the substance and help it to move across the membrane. They will actually combine with the substance. They're carriers. So you can think of carry the substance across. Versus channel protein. The channel protein is just an open channel. Says, all right, go. Okay, so carrier proteins. Next one we have cell recognition proteins. Now when we talk cell recognition proteins, they help the body recognize when it is being invaded by pathogens. Here they're giving us an example of MHC. MHC is major histocompatibility complex. The MHC glycoproteins are going to be different, we're going to see, from person to person. So when we talk organ transplants, so organ transplants are difficult to achieve because we'll see cells with foreign MHC glycoproteins are attacked. They're attacked by white blood cells that are going to be responsible for immunity. So that's why we have to have a match. Next, then we have receptor proteins. Receptor proteins, they have a shape that's going to allow a specific molecule only to bind to it. The binding of this molecule causes the proteins to change in shape. And when they change in shape, this causes a cell response. So here we can see we've got a protein, and this protein has a specific shape to it. So if there is, you can see here now, we have a molecule that has the same shape as this receptor. Now, if it didn't have the same shape as this receptor, let's say this molecule was, triangular shaped. Okay, let's say it was triangular shaped, it wouldn't be able to fit in here. And it wouldn't be able to elicit a response from the cell then. It's like saying we have keys, right? We have keys. However, my keys don't open your house. Your keys don't open my house, but we do have keys. So they're molecules. However, we have to have the right molecule, or even garage door openers. The garage door openers, right? We all have one, but again, mine only opens my garage. It's programmed to open mine. Yours is programmed to open yours. Okay, so that's kind of the same thing. It's a lock and key type of mechanism there with the receptor proteins. And then we also have enzymatic proteins. Now, enzymatic proteins are going to carry out metabolic reactions directly. They carry out metabolic reactions directly. They can catalyze specific reactions. Reactions that would take a little bit longer to occur come into contact here with this enzymatic protein, and they're going to occur almost immediately. And we're going to go through and we're going to check that out as we progress through the rest of the semester. Next, let's talk about the permeability of the plasma membrane. Let's talk about the permeability of the plasma membrane. Now, the plasma membrane regulates the passage of molecules into and out of the cell we've described because the membrane, this plasma membrane, regulates the passage of molecules into and out of the cell. This plasma membrane is what we call selectively permeable. It is selectively permeable, meaning that it allows certain substances to move across the membrane while not allowing others. So it is selectively permeable. Some molecules are going to be able to freely cross that plasma membrane, the phospholipid layer. And other substances may require transport by certain proteins that we talked about, carrier proteins or channel proteins, and even energy may or may not be used. So that plasma membrane is a selectively permeable barrier. Now I want you to know small, non-charged molecules like carbon dioxide, oxygen, alcohol, they can freely cross the membrane through the phospholipids. I'm happy with alcohol, right? So we can have the effects a little bit faster. Otherwise, it would take time. You have to get ATP, move it across, or I don't know what other mechanism would be involved there. Just to make this fun, okay? And add some fun there too. Okay, so again, small, non-charged molecules like carbon dioxide, oxygen, and alcohol are able to cross freely through the membrane, through the phospholipids. So here we can see them. They're able to slip right in between those hydrophilic heads and pass through those hydrophobic tails and make their way right into the cell. And these few examples you see here, we're going to see they are said to go down their concentration gradients. They are said to go down their concentration gradients when they move from an area where their concentration is higher in quantity compared to an area where their concentration is lower in quantity. So they are driven by their concentration gradient. They move from areas of high quantity to areas of low quantity. That's very good for oxygen and carbon dioxide. Right? Because every time we breathe in and out, we are bringing in oxygen and removing CO2. So the driving force in the tissue we see is quantities. Oxygen quantity. Oxygen is going to move now from the air into the lungs, from the lungs then into the blood, thanks to concentration gradient difference. Okay, so we said these molecules are they're driven by their concentration gradient. Okay, they're going to be moving from an area where they're going to be found in higher concentrations to where they're found in lower concentrations. I want you to know that there are also some molecules that are going to be able to go up their concentration gradient, not down their concentration gradient like we just saw here, but up their concentration gradients. Now, when we say that they're moving up their concentration gradients, we're seeing that they're going to be moving from an area where their concentration is actually low to an area where their concentration is going to be high. Now, this type of movement is going to require energy. It will require energy, which is going to be found in the form of ATP in our bodies. Now, when we talk water, okay, so here you can see substances can be found going up and going down their concentration gradients. So when they're going down a concentration gradient, they're moving from an area of high to low. Going up, they're going from an area of low to high. And this will require energy, as we said. So this is how substances can move across that membrane, either going up concentration gradient or going down a concentration gradient. Next then, we'll talk water. Water, I want you guys to know, is going to be a polar molecule. Water is a polar molecule, and it cannot penetrate the non-polar membrane. Water is polar, which would not be expected to readily cross the membrane. Most cells have special channel proteins that are called aquaporins. I like to think of them as agua in Spanish, water, aqua, porins. Even aqua is pertaining to water, right? Aqua sports or aqua clothing or whatever you have there. So when we talk aquaporins are going to allow water to cross. Special channels that are going to allow water to cross the membrane. Now, aquaporins are going to be present in the majority of cells. They'll be present in the majority of cells. Now, when we talk large molecules, some ions, and charged molecules, we'll see they are unable to freely cross the membrane. They are unable to freely cross the membrane. They require channel proteins. They require channel proteins, carrier proteins, or even vesicles. So large molecules, some ions, and charged molecules. They cannot just freely cross the membrane. How do they get across the membrane then? They're able to get across the membrane thanks to channel proteins. Channel proteins, they form a pore through the membrane which will allow these substances to make their way through. Carrier proteins are there as well that'll help. Carrier proteins are going to be specific for substances. They don't allow anything and everything to pass through. They're going to be specific for substances, and they'll allow them to then make their way across, as we saw. They'll carry them across. Now, vesicle formation. Vesicle formation, we're going to see in the process of endocytosis and also exocytosis. Endocytosis is going to be the process of bringing the substance into the cell via a vesicle. Exocytosis is removing it from the cell through a vesicle. So vesicle formation in endo and exocytoses. So here we can see aquaporins. If those substances can't just freely cross through the phospholipid layer, like carbon dioxide, oxygen, and certain alcohols, then, like water, special carrier, okay, macromolecules you see here, they can't just penetrate through. Charged molecules and certain ions can't just make their way through. You have this nonpolar hydrophobic core. It's not going to allow these substances right through. So then you've got those specific channels and carriers or vesicles we're going to go through, we're going to see as well. And these vesicles, they're going to be made, okay, because here you know these are proteins. Channel proteins, carrier proteins, vesicles, you're probably thinking, what is that? Vesicles are going to be made up of these phospholipids. Now, whenever this big macromolecule pushes up against this phospholipid bilayer, this phospholipid bilayer will kind of pinch off and surround it. And then it'll close itself back up, and this molecule is going to make its way inside. It'll be in that vesicle. And then you saw lysosomes can come and greet it. Hey, what are you doing here? Why are you here? What's going on? And, and kind of pursue it and see what's going on and take care of it if it's an invader. So our body's, if it's not already getting fascinating, it's going to get even more exciting. So it's just these barriers we have there with our cells. Now, when we go through and we talk about the passage of molecules into and out of the cell, what we're going to do is we're going to go through and we're going to see a couple of different processes. We're going to see a few different processes. We talked endo and exocytosis, so we're going to check those out as well. And what we're going to do is we're going to divide these processes up into processes that require energy and don't require energy. So let's go through and let's talk diffusion and osmosis first. Let's talk diffusion and osmosis. We talk diffusion. Diffusion is the movement of molecules from an area of higher concentration to an area of lower concentration. It's the movement of molecules from an area of higher concentration to an area of lower concentration. Again, this is movement down their concentration gradient. So we are moving down their concentration gradient whenever we're going from high to low. So you can think of it as, let's say we're inside of a classroom or you're inside your bedroom, somebody comes to visit you and they open the door and they're wearing some type of cologne or perfume. And, you're sitting across the room and all of a sudden after they open the door, a minute later, you smell them and, you say, oh, wow, this smells really nice. You see what's actually happening there is you have diffusion taking place. These molecules are found in a higher concentration on this individual that opens the door and comes in. And then all of a sudden, they're going to diffuse. They're going to diffuse to an area where they're in lower concentrations, which is going to be inside of that room. And so as they diffuse across the room, then they'll make their way to your nose, and you're going to smell and you'll know exactly what's happening there. So it's a movement of molecules from an area of higher concentration to an area of lower concentration. That's just one example I'm giving you there. And this can be observed, we'll see. Okay, and the movement of these molecules is going to keep happening until equilibrium is reached, until they are spread out in a nice fashion, until equilibrium is reached. For example, we're going to see here crystal dye. When crystal dye is going to get placed in water, the dye molecules are going to start moving around. They're going to start moving around until there's equilibrium and the water becomes purple. Okay, if the crystal dye is purple that we're using. So we can see that right in here. Here we've got nice little poor fishies. I hope it's non-toxic. I hope it's not toxic, non-toxic and doesn't harm them. But here you can see this crystal dye. The crystal dye is introduced to this fish tank here. And here the crystal dye is found in higher quantities here in this area here, as we can see, compared to out here. So what happens is it starts to move around from areas of high concentration to areas of low concentration until equilibrium is reached, until it's found scattered just about everywhere. And you can see here the whole water, this whole fish tank becomes purple then. All the water becomes purple. All the water in the fish tank becomes purple. So this is diffusion. This is diffusion. So this can be observed when crystal dye is placed. You can see here in water, the dye and the water molecules are going to move in various directions. The water molecules aren't found in high concentration here. They're going to be making their way into there. And so they're doing that. They're coming in here. The dye is moving that way until you have an equilibrium reached. So they're all moving in various directions. Their net movement is towards the region of lower concentration. Eventually, the dye is dissolved in the water, resulting in an equilibrium and a colored solution, as we see here. Now, when we talk solutions, so this solution you see here, when we talk solutions, we're going to eventually get to IV solutions, what we're trying to put the big picture towards. So when we talk solutions, I want you to know a solution is going to contain both a solute, which in this case is the dye, a solute. In this case was a dye, so it's a solid, and a solvent. And a solvent. And the solvent is usually a liquid, as we see here. So the solute is a dye, solvent is a liquid. So solutions usually contain a solute and a solvent. Next then let's talk. So here you can see how oxygen, okay, and CO2 did the opposite, is able to move across from inside the lungs into the bloodstream and move from an area where it's found in high concentration to an area of low concentration. So the same thing we're seeing here. And if we had CO2, CO2 would be inside of here and it would be moving in the opposite direction. So it could be expelled out. Next then, several factors I'd like you to know are going to influence the rate of diffusion. Temperature is one important factor. As temperature increases, the rate of diffusion increases. Pressure is also going to influence the rate of diffusion, as well electrical currents, and the size of the molecules, molecular size. All of these factors influence the rate of diffusion. Next is then the term osmosis I'd like you to know about. When we use the term osmosis, now osmosis is defined, you can see, as the diffusion of water. 1, 2, 3, 4 words. The diffusion of water. Because you've got to understand what the means. Obviously we know. Diffusion, obviously now we know the movement from high to low, right? From high concentration to low concentration. The diffusion of, we know what that means, water. 4 words. So the movement of water from a high concentration to low concentration. So osmosis, diffusion of water across a differentially permeable membrane. And this diffusion always occurs from high to low. So if somebody asks you, okay, when you get to anatomy, the next class, they ask you, can you define osmosis? The diffusion of water. Okay, they should understand that you know exactly what you're talking about there then. Okay. Now, when we go through and we talk the diffusion of water, so it's again the diffusion of water across a selectively permeable membrane. The diffusion of water across a selectively permeable membrane due to concentration differences, right? Due to concentration differences. Due to concentration differences. Now, osmotic pressure is the pressure that develops in a system due to osmosis. Now, the greater the possible osmotic pressure, the more likely it is that the water will diffuse in that direction. Here you can see this example. Now here in this example, we have a tube containing a 10% solute solution we see right in here. It has a 10% solute concentration. So this substance here in green, okay, is going to have a 10% solute concentration versus this fluid here in the beaker, which is a lower percentage of solute. It's only 5%. So where do we have more solutes and solvent concentration? So in this tube, we have a higher solute to water concentration versus in the beaker. So we're comparing those two environments. So here now, a tube containing this 10% solute concentration, you can see is covered at one end, and it's covered at this end by a selectively permeable membrane. So we get this tube that's covered on one end with the selectively permeable membrane and filled with a 10% solute concentration solution. And we submerge it. We place it into a 5% solute solution beaker. Now again, the beaker has a higher concentration of water molecules, lower percentage of solutes, and the tube has a lower concentration of water molecules and a higher percentage of solutes. Diffusion always occurs from higher concentration to lower concentration we described. So a net movement of water takes place across the membrane, and it moves from the beaker to the inside of the tube. The solutes do not diffuse out of the tube. A net movement of the water takes place across the membrane from the beaker to the inside of the tube. To the inside of the tube. The solute does not diffuse out of the tube. The solute does not diffuse out of the tube because the membrane is not permeable to the solute. It's selectively permeable, so selectively permeable to just the water. Now in the end, the concentration of solute in the tube, you can see, becomes less than 10%. Becomes less than 10% because the water moved up into the tube, and before it was at 10%. Now how much water we moved in, we haven't calculated exactly, but we know now, look at the volume was only up to here. The volume has increased to the top. So that 10% has been diluted. It's less than 10% now. In the end, the concentration of solute in the tube is less than 10% because there is now less solute per volume of solution. So the membrane is not permeable. It's selectively permeable. It's not permeable to the dye, to the solutes. It's not permeable to the solutes. It is permeable to the water though. So water moves across. Why is water moving across? Because there's a less concentration of water inside of this tube compared to out here in the beaker. So water moves from high concentration to a low concentration. What do we call that? Osmosis. So here you can see osmosis took place. There was a diffusion of water across the selectively permeable membrane because there was a concentration difference. Now, if I told you that this selectively permeable barrier, the selectively permeable membrane, is also going to be permeable to the solute, so then the solute is going to move from a high to a low. It'll move from the 10% down into the 5%. Okay? All right, next then, let's talk about different solutions. Let's see what solutions can do to our cells. Now, when we go through and we talk about different solutions, I'd like you guys to know first about an isotonic solution. Now, isotonic solutions in the lab, we'll see first. Now, in the lab, cells are going to be placed in isotonic solutions. This is the same thing as you would be doing with IV solutions. Okay, now first I'd like you to know that the prefix iso, this prefix iso means the same as, means same as. Now the term tonicity, tonicity refers to the osmotic pressure of the solution. It refers to the osmotic pressure of the solution. Now in an isotonic solution, the solute concentration in an isotonic solution, the solute concentration and the water concentration, both inside and outside the cell, are equal, and therefore there is no net gain or loss of water from the cell. Now, a 0.9% solution of the salt sodium chloride, that is known to be isotonic to our red blood cells. 0.9% sodium chloride solution is known to be isotonic to our red blood cells. IV solutions, medically administered, usually have this tonicity. And again, it all depends on what you need. They have all different types of tonicities. Let's talk then hypotonic solutions. Hypotonic solutions are going to be solutions that cause cells to swell, or even burst. That happens due to an intake of water. And they're said to be, that's due to an intake of water in or from those hypotonic solutions. Now the prefix hypo means less than. It means less than, and it refers to a solution with a lower concentration of solute than the inside of the cell. So if a cell is placed in a hypotonic solution, water enters the cell. If a salt solution is lower than 0.9%, we consider it to be hypotonic. It would be hypotonic to our red blood cells. So let's look at a picture. So here we can see, if we were to get this cell, okay, this animal cell we see here, this red blood cell we see here in this picture, and we were to place it in an isotonic solution, a solution that has the same tonicity as the inside of the cell. So equal inside and outside, you're going to see there's no net gain or net loss of water by the cell. Now, if we were to get that same cell, again, place it in a hypotonic solution, a solution that has a tonicity less than 0.9%. So let's say 0.5%, 0.4% compared to the inside of the cell. That's going to cause the cell then to pick up more water, and eventually it can burst, or lysis can take place. Now, if we were to get a cell and place the cell in a hypertonic solution, a hypertonic solution is a solution that causes the cells to shrink or shrivel due to a loss of water. The prefix hyper means more than, and it refers to a solution with a higher percentage of solutes than the cell. So if a cell is placed in a hypertonic solution, you can see water leaves the cell. Okay, water leaves the cell and it moves into the outside. So it leaves the cell, and you will have the cell begin to shrivel, or you say crenation. takes place. Crenation takes place. So these are three situations that can happen in relation to the cell being placed in different concentration solutions. Here's the same thing to a plant cell. A plant cell. Now, plant cell, when it gets placed in an isotonic solution, you can see again, nothing happens. Okay, same amount of water that's gained is going to be lost. Now here you can see in a hypotonic solution. Now you'll understand why I said I wish we had plants. I wish we had cell walls. Here you can see now in a plant cell, eukaryotic cell, in a plant cell, when it undergoes, when it's placed in a hypotonic solution, the cell gains water. Here now, what's going to happen is turgor pressure is going to keep the plants erect because of the cell wall. And also, if needed, this vacuole, the central vacuole, will start to fill up with water also. So here you can see, if we're to get basically a leaf and place it underneath the microscope, you can see what would happen to the cell walls. Here you can see what happens to the plant cells. Now here you can see what happens to the plant cell if we place it in hypotonic solution. So you can see here the vacuole picks up with water. Vacuole picks up all the water. And then you can see the chloroplast and everything else is going to move off to the sides. So that's exactly what you're seeing around here. It doesn't burst. The cell walls are going to keep the cell from bursting. Now here, if we were to place it in a hypertonic solution, you can see here now, what happens is the cell, its plasma membrane, is going to all start to shrivel to the inside. So in a hypertonic solution, you can see what happens to the cell, the plant cell. The central vacuole loses water, and the cytoplasm shrinks. You have what we call plasmolysis takes place. And the chloroplasts are even going to be seen right in the center. You can see all that right there in the picture too. Now let's talk about transport by carrier proteins. When we talk about transport by carrier proteins, I'd like you to know carrier proteins are specific as each combines with only a certain type of molecule. So we talk transport by carrier proteins. Carrier proteins are specific. They're specific as each combines with only a certain type of molecule or ion, which then gets transported through the membrane. So here we're going to be talking about how substances are going to enter or exit the cell because of carrier proteins. And carrier proteins, we said, are specific. We're going to see they change shape. Their shape changes. In order for them to be able to move the molecules across the membrane, they have to have a shape change take place. So now when we go through and we talk about transport by carrier proteins are required for facilitated transport and for active transport. Let's talk first facilitated transport. When we talk facilitated transport explains the passage of molecules like glucose and amino acids. It is going to explain the passage of molecules like glucose and amino acids. Facilitated transport explains the passage of molecules such as glucose and amino acids across this plasma membrane. Here, the substances combine with the carrier proteins, and then they're transported across the membrane. This process and diffusion, we said earlier, they do not require ATP. They don't require ATP because the molecules are going to be moving down their concentration gradient. So here you can see we have the solutes that we'd like to move across this membrane, move them to the inside of the cell, and here's the carrier protein. So the carrier protein looks like this, and then we said it's going to carry the solute that it wants to move, and so you can see here now the shape is changed, and then it allows the substance to make its way in, and then it resets. And here again, energy is not required. Concentration gradient is the driving force. Next, we have active transport. When we talk active transport, this is going to require basically ATP we're going to see. So carrier proteins and energy. Carrier proteins and ATP are required to transport molecules against their concentration gradients. Against their concentration gradients. Proteins involved in active transport are usually called pumps. They're often called pumps. We'll talk a lot about the sodium-potassium pumps as we go through the semester. And here we can actually see sodium-potassium pump. So here the sodium-potassium pump is going to allow sodium and potassium to move to their respective regions thanks to a carrier and energy. Thanks to carrier energy. So when we talk active transport, molecules or ions are going to combine with the carrier proteins that are often called pumps. In active transport, so the molecules or ions are going to combine with the carrier proteins that are often called pumps. These molecules, they move against the concentration gradients, entering or leaving the cell, and they accumulate either inside or outside the cell. So then energy and carrier proteins are going to be required to help move them to their respective sides. And it's usually ATP that's going to be used, our energy. Proteins in active transport are referred to as pumps, as we said. So proteins are going to use energy to move molecules against the concentration gradient. The sodium-potassium pump is very important. It's especially important for nerve and muscle cells. And what it does, it moves sodium out of the cell and potassium back into the cell. So it moves sodium back out of the cell and it moves sodium back out of the cell and it moves potassium back into the cells. The carrier changes shape after phosphate attaches to it and then again after it detaches. So here you can see this is the shape of the original carrier. And you can see there's potassium on the outside. Potassium leaked and it made its way out. Sodium leaked and made its way in. So we have to move them to their respective sides. So this pump here accommodates 3 sodium, you can see, and 2 potassium. So when 3 sodium come and sit in here, it's charged by ATP, it's ready to move them, and then it'll grab 2 potassium and bring them inside. So here, let's look at that happen. So 3 sodiums assume their seat. It becomes phosphorylated. This ATP gives up its phosphate, becomes ATP, and now this is, you can think of it's charged. This pump is charged, and it's ready to elicit this action. So now here what happens is, look at the shape changes. Sodium makes its way out, okay? And then here we're going to see potassium are going to take their seats while the door is open on that side. So potassium takes its seat, and then potassium is going to be closed. You can see the energy got used, and then the pump is going to reset, allowing potassium to make its way in. So very important pump we're going to see again in relation to nerve cells and muscle cells. Next, then we talk about bulk transport. Now, I'd like you to know macromolecules are going to be transported into or out of the cells by vesicle formation. So macromolecules are molecules that are too large to be transported by carrier proteins. Macro, big molecules. Energy is required to form the vesicles. Now, vesicle formation is called membrane-assisted transport. And we have either exocytosis, where we move things out of the cell, or endocytosis, where substances are going to be moving into the cell. So energy is required to form vesicles, and vesicle formation is called membrane-assisted transport, where we have two types: exocytosis, where it'll exit from the cell, and endocytosis, where substances will enter, macromolecules will enter into the cell. So let's look at that. First, we'll talk exocytosis. Exocytosis, the vesicles are going to fuse with the plasma membrane as secretion occurs. So as the substances are going to move out, right, the vesicle is going to fuse with the plasma membrane. The vesicle membrane becomes part of the plasma membrane, because I told you the vesicle is formed from the plasma membrane. Cells of particular organs are specialized to produce and export molecules. Pancreatic cells, for example, they release insulin or enzymes. And our anterior pituitary, the anterior pituitary cells release growth hormone. So here we can see, let's say, here we have the vesicle. The vesicle is moving out towards the plasma membrane, out of the cell. So when it moves towards the plasma membrane, you can see here then, it makes its way closer to the plasma membrane, and eventually those phospholipids all start to fuse together. And then here you can see the substances that were moving out are able to make their way out. And then the vesicle will fuse right into the plasma membrane. Let's look at the same exact picture for endocytosis then, moving in. Now moving in, we'll see the macromolecules are going to come, they're going to basically push up against the plasma membrane, causing that plasma membrane to pinch off. So here you can see they'll get enclosed by this vesicle that's made by the plasma membrane, and it'll pinch off. And it pinches off the plasma membrane, and it makes its way into the cell. So you can use that same picture for both processes. We can look at it this way for exocytosis, and we look at it this way for endocytosis. So endocytosis, cell takes in substances by vesicle formation. What happens? Part of the plasma membrane invaginates to envelope the substance, as we just saw. The membrane then pinches off to form an intracellular vesicle. When we talk endocytosis, we have three types of endocytosis. We have first what we call phagocytosis, pinocytosis, and receptor-mediated endocytosis. So three different types of endocytosis. Phagocytosis is a process that engulfs large molecules. It's a process that engulfs large molecules, particulate matter such as food molecules, or viruses, or even whole cells, like with amoebas and macrophages. Pinocytosis, you'll see liquids and small particles dissolved in liquid are going to be making their way in. Certain blood cells or plant root cells are capable of doing that. And then receptor-mediated endocytosis is a type of pinocytosis. Receptor-mediated endocytosis is a type of pinocytosis that involves a coated pit. It involves a coated pit. And certain placental cells are able to exhibit receptor-mediated endocytosis. So here you can see phagocytosis. And here we have pinocytosis. And here we have receptor-mediated endocytosis. Next, let's move down to 4.3. 4.3 takes us into modifications of cell surfaces. Now, when we talk modifications of cell surfaces, we'll see here most cells interact with other cells and their environments. Cells live and interact with their external environment. They don't live isolated. We'll speak of two features. Number one, we'll see the extracellular matrix, which is outside the cells. And number two, junctions we're going to talk about that are going to be found occurring between some types of cells. So here we're going to see the extracellular environment is made of large molecules, and they're going to be produced by nearby cells. And the materials are deposited by secretion. Plants, prokaryotes, fungi, they're surrounded by cell walls. And animals have more varied extracellular environments that can change. So that's what we're really honing in on. So animal surfaces, animal cells have two different types of cell surfaces. Extracellular matrix, which is again on the outside, and then junctions that are going to occur between cells. Now, both can associate, both can associate with the cytoskeleton and can contribute to cell communication. So first is the extracellular matrix. The extracellular matrix is found outside the cell, and it is a meshwork of proteins and polysaccharides, which is in close association with the cell. It also contains collagen. It also contains collagen and elastin fibers, which are structural proteins, So common structural proteins in the extracellular matrix, you can see collagen, because it resists stretching. Elastin, it provides resilience to the extracellular matrix. And then fibronectin. Fibronectin is an adhesive protein that helps to link integrin. Polysaccharides made of amino sugars in extracellular matrix are going to attach to proteins called proteoglycans. Proteoglycans attach to a long, centrally placed polysaccharide. And this is going to help resist compression of the extracellular matrix. And it assists cell signaling by regulating the passage of molecules through the extracellular matrix to the plasma membrane. This is all sophisticated. Then the junctions between the cells. You'll see cell surfaces in certain tissues of animal cells. Let's talk about junctions between cells. These junctions are going to be found on the cell surfaces in certain tissues of animals. Now, junctions between cells include adhesion junctions. Adhesion junctions are going to be intracellular filaments that are found between cells. Then you also have tight junctions. Tight junctions, they form impermeable barriers between cells. And then third are gap junctions. You're going to see gap junctions are going to be junctions where the plasma membrane cells are joined. And this allows for communication. We're going to see this in relation to cardiac muscle cells. We're going to see this in relation to smooth muscle cells also. So here you can see that extracellular matrix on the outside. And here's the plasma membrane. And then certain proteins that are going to be attached to it on this side. And then attached to actin filaments you can see on the inside. So all doing various functions. Here we can appreciate adhesion junctions. Here we have tight junctions. Okay, you see the difference between the two? And then here you have gap junctions, where there's actual communication between the two cells.